All right, so my name is Adrian Skobelski. Today is November 3rd, 2022, and my presentation is on the anatomy and physiology of the retina. All right, so let's get going with this. All right, so here's my presentation, the anatomy and physiology of the retina. Okay, so to start off, the retina is the major part of the eye. It is the light-sensitive lining that is in the back of the eye, as you can see in the depiction. There encompasses 5 different types of neurons. There's photoreceptors, bipolar cells, ganglion cells, amacrine cells, and horizontal cells. So today the primary focus will be on the photoreceptors, which play an important role in how we are able to see and how valuable it is in terms of the optical pathway. And so on the side over here you have a clear image of how the eye looks like, and we're going to focus on the retina today. Alright, so as I mentioned, today we're going to be focusing on the photoreceptors, and the retina contains 2 major types of photoreceptors. They are the rods and cones, and what they are known for, well, rods are good for seeing in low light settings, and they definitely outnumber the cones in the retina. And while we are able to use cones for higher levels of light, and the cones are responsible for helping us see color, there's a whole different process on what that involves in terms of seeing different wavelengths of light and all that stuff. But here in this presentation, we see a clear depiction of the orientation of the retina and the different components. So as I mentioned in the previous slide, there are 5 major types of neurons. And so we see how they're organized in this picture. We have on the bottom, we have the rod cell, the cone, the ganglion, bipolar, amacrine, and the horizontal cells. And in this picture, you see how it's oriented. If we go from the left side, which is the inner membrane, all the way to the right, we start with nerve fiber, which connects to the ganglion, which has the ganglion cells, which connect to the bipolar cells, which then leads to the back, which are the photoreceptors, which is very interesting because you would expect photoreceptors to be on the inner part of the membrane, but in the retina, the photoreceptors are actually in the back. And so what happens is, as light goes through the lens and comes into the retina, we'll see how stimulating the photoreceptors plays an important role in how we are able to see images. Okay, which brings me to another classic point. As I mentioned earlier, the rods heavily outnumber cones, but there's an important spot on the retina called the fovea. Now in the fovea, this is where there's a high concentration of cones, and it looks like a little dip, as you can see in the depiction, where there's no other neurons, it's just straight focal point onto cone photoreceptors. Now, why is this important? Well, we know that cones play an important part of seeing color, and they're known for helping us see with high visual acuity. And so we need this fovea in order to process precise, complete activities that require precise vision. You'll need it for driving, for reading. Really an important part of the retina, and the depiction shows how there's no disruption in how the light could go through, and the light goes straight into the cones and stimulates those photoreceptors. Alright, so we got through the anatomy part of the retina, and now we're going to go into the physiology part of the retina, which leads to the question on what leads to the stimulation of these photoreceptors. Okay, well, first we're going to talk about phototransduction during dark phases. So in my examples, we're going to talk about phototransduction in the rods, but this is a very similar process that occurs in the cones. Okay, so just like in any other part of physiology, we have a bunch of ion channels, and in the dark, you have, well, in the dark or the light, there's three types of channels. We have CNG channels, potassium channels, calcium channels. Alright, and so when it's dark, you can see in the depiction, there's a large concentration of what looks like these hexagonal molecules, and what that is called cyclic guanosine monophosphates. And so what happens is, and we'll use the short term CGMP, and so when we have high levels of these CGMP molecules, this allows for these ion channels to be open, and so positively charged sodium and calcium ions are able to come into the membrane, leading to a depolarization of the cell, and it goes from about negative 70 millivolts to negative 40 millivolts. So what happens is, as you can see on the bottom part of the picture, we have a slight depolarization that occurs in the rod. And what happens is, as those sodium ions come in, the calcium ions come in, we have a neurotransmitter that's released because of this depolarization. And what that neurotransmitter is called glutamate. So on the bottom, you can see glutamate molecules are being released, and we have a depolarized cell, glutamate's released. I'll talk about the importance of what happens when that neurotransmitter is released. But in the dark phase, a protein that's very valuable in the light phase is inactive, which allows all of this to continue to happen. And that protein is called rhodopsin, which is made out of opsin and retinol. Okay. All right. So then, what happens during phototransduction when there's light? So, as I mentioned, the protein rhodopsin is made out of both opsin and retinol. Alright, so I'm going to go through this picture and kind of demonstrate what happens. So, as light goes through the lens and hits these photoreceptors to stimulate it, the light is actually absorbed by retinol, which leads to a little chain reaction. So what it does is, it leads to opsin to activate another molecule protein called transducin. And what happens is transducin activates another enzyme called phosphodiesterase. So what is the purpose of phosphodiesterase? Well, phosphodiesterase is an enzyme, and what it does, it works to lower the CGMP levels. And so the CGMP, the cyclic guanosine monophosphates, in the dark phase, why they were important is they allowed ion channels to remain open, which allowed the depolarization to continue. But when we have this phosphodiesterase activated, what happens is the CGMP levels go down, and that closes the important ion channels, so sodium can no longer flow in. So what happens is, as the potassium ions continue to flow out, this leads to a hyperpolarization of the cell. And as you can see on the bottom, we have a hyperpolarization. Potassium continues to flow out, and there's no flow of positive charge from the outside of the cell. So what happens is, that neurotransmitter glutamate is no longer released. So that glutamate is, as you can see in the picture, is stopped, it's not released, and this is when light stimulates the cell. So light hits the retinol is absorbed, turns on transducin, which activates phosphodiesterase, takes down CGMP levels, leads to a hyperpolarization of the cell. Alright, so now we get to the important part of why glutamate is important. So we're going to start with the dark phase. As we know, the CGMP levels remain normal, glutamate is released, and now it leads to the stimulation of bipolar cells. And as we know, bipolar cells connected to from the photoreceptors, connected to the ganglion cells, which then lead to the optic nerve. So when it's dark, there are two types of bipolar cells. We have the on bipolar cells, which means light on, and we have bipolar cells which are called off bipolar cells. And in the dark phase, when glutamate is released, the light off bipolar cells are excited while the on bipolar cells are inhibited. And so what that means is that ganglion cells will be inhibited. And when we turn to the phase when light occurs and it hits the retina, we have glutamate that is not released. cGMP levels are decreased by the phosphodiesterase. Glutamate neurotransmitter is not released, but we still have on bipolar cells that are excited and off bipolar cells that are inhibited. So this is important because what this means is that glutamate, although it's one neurotransmitter, can have different effects based on whether it's released or whether it's not released. And so we can see the comparison between what happens in the dark and what happens in the light. Because normally when we have a specific neurotransmitter, we know that neurotransmitter just works on a specific reaction. But here it could be released or not released and still have an effect on how we see. And then following what happens is there's a whole complex machinery which happens to restore the retinal, to turn the rod back to its original phase, and that's after the light phase. So that is my presentation on the anatomy and physiology of the retina. I hope you enjoyed it and hope I was able to describe everything as well as possible. Thank you.